Hello everyone, and welcome to the tutorial session of Functional Genomics. Myself Anupama, and as already introduced, I am one of the teaching assistants in this course, that is Functional Genomics. Today we will be learning about what all informations can be arrived at or fetched using few hundred base pairs of DNA sequence. So now let's look into that. So this is a part of chromosome which was deleted in a disease and linked to that disease. This was identified by a team of scientists. So few of you must be wondering that how did they arrived at the region of the chromosomes. So that has been covered already in one of the NEPTL course, that is Human Molecular Genetics, and also we are rerunning that course. But since you are here, very briefly I will like to tell you that how do we arrived at such sequences. So what is done, that there are families which come for counseling to the geneticists, and they have members who are affected. Suppose this female was affected and she has another sister, and their parents were unaffected. Similarly, there was another family, family 2, family 3 with family 4. So once you have a certain amount of family for which you can screen for the cause of the disease. So what is done, that DNA is extracted from all the family members which are present, and then there are EST markers. Now what are EST markers? These are small expressed sequence tag which are unique for particular region of the chromosome. These are unique for the regions. So what do the scientists do? They screen for these EST sequence. So suppose approximately around 250 EST sequence can cover almost all the autosomes of the chromosomes, and from there on they try to see that what EST region or what EST is getting segregated with the disease. By that I mean that which one are always present in the individuals which are affected. For example, in this female, and then the other person of the other family which is affected, if that EST marker is present or not. So from that they narrow down to the region of the chromosome, and in this disease the case was the chromosome 6 and this region. So from here, what using this EST and a small probe they design, and then they screen cDNA libraries for that. They try to align them as they screen for many libraries, sequence them as sequencing has already been taught, and there are various ways by which you can sequence, and then you arrive at a sequence. In this session we will start with the sequence which has been arrived, and these are few hundred base pair of the sequence. This is the 5' end and this is the 3' end of the sequence. So using the current search tools, what all informations we can take is we can arrive at, or which can be fetched using this few hundred base pair of the sequence. That's what we will be learning today. So there are few widely used search engines. One of those is NCBI, another one is UCSC Genome Browser, another one is Ensemble. So today we will be learning about more about NCBI, and then we will look online on another search engine, that is Ensemble. So since we had a few hundred base pair of sequence and we didn't know from which gene it is, or whether this is a coding region or not coding region, or this is just an intron of a gene, or this is an exon of a gene, all those informations we don't have. So one such way is to use BLAST, and here is the page of BLAST, that is basic local alignment search tool. So what does BLAST does? It finds the region of similarity between biological sequences. This program, it compares the nucleotides or protein sequences to sequence database and calculates the statistical significance. So there are various BLAST programs, like one is nucleotide BLAST, the other one is protein BLAST. Now you have a stretch of a sequence that can be converted into protein, and then again it can be BLAST to the protein database, that is called BLASTX. Similarly, the protein can be translated to nucleotide, and then it can be BLAST to the other nucleotide database, and that is called tBLASTn. So as we have seen that we had a DNA sequence, so we will go for nucleotide BLAST and try to look at what region it is aligning to. But before we go into what region it is aligning to, very briefly we will look into what does sequence alignment means, and what are the various strategies used to align the sequence. So there are two key terms which you will encounter more often, that is global versus local alignment. So very briefly, what is global and what is local alignment? Let's look into these two sequences. Now, one, you have to align these two sequences. One is this sequence, and the other one is this. Now, there are two ways by which you can align this sequence. One is that you just go one by one and try and align. For example, here if I align this one with this, so this would be T, A, C, T, C, A, and so on, and you will try and calculate the score, which is based on the various algorithms, that what is the score of this alignment. Now another way is to that you you don't go one by one, but you try and look for small stretch which are aligning the best. For example, here it would be if you align here with this T, A, C, T, C, A, C, G, G, and so on, you will see that the local stretches are aligning more. So this is called global alignment, the first method, and the other method is called the local alignment. Similarly, if you have to align these two sequences which are almost of same length, then it is best to align them globally. So when there is a small stretch of sequence, it's best to align them local, and if there are equal length of DNA sequences, then that can be aligned globally. So on these alignments, using different algorithms, scores are calculated. A few of those is like from where you are starting, what is the gap between the first not aligned and the one where the alignment starts, there is a gap penalty. Then in between there are penalties where there are mismatches, or you skip few bases and then you align to the another stretch. For example, suppose this is one of the sequence, this is 5 prime 3 prime, and then you have another sequence, so it aligns here, but then there are some mismatches, so you skip that and then again you align. So there would be a certain penalty for this, and then that penalty would increase at these regions. So on basis of these, the score is calculated, and that is also represented in our BLAST data. So now let's look into how do we go about it. So what happens, like this is the BLAST homepage where you have different different ways, like as I have already described, BLAST N, BLAST P for protein, BLAST X where nucleotide is converted to the protein and then BLAST to the whole protein database. Similarly, the nucleotide is translated and then it is blasted to or aligned to the sequences in the protein database, and so on. So here you can enter your sequence, which has that sequence can be entered in a different formats, which has accession numbers which are given by NCBI, and that is are the unique. Suppose you have a gene for which you want to blast, you just need to put that accession number and the tool would take it, or you can paste FASTA sequence. Now FASTA sequence is a format of writing the nucleotide or the DNA sequences. Then on the basis of your requirement, you can make the, you can use a particular database. Suppose you just want to use the human genomic or the transcript or mouse genomic transcript, or you can choose from the other organisms or exclude them, add them, whatever is your criteria, you can do that. And then you can just click to blast. So in our case, this was the query sequence which I have pasted here, and then you click on blast. Just this is the default setting, I haven't changed anything, and you click on blast. So what do you get the results? Sometimes the software takes time in doing that, and this is how the page it comes. This is the job title, and this is your query sequence, and this is the time you submitted and how much time duration has passed on. And then you have a result page. This is the blast result page. So here are the this is the query, the description. You had put in nucleic acid which was 421 base pair long, and this is the database through which it has done the search, and that is non-redundant database. Non-redundant database. Then it has the description is the nucleotide collections, then this is blast n, and then you get a distribution of the top 40 blasts on the 40 subject sequence. So these are the 40 sequence subject sequences against which these are the best scored ones, and the score has been here shown in the format of color. So red shows the highest one, and the black shows the lowest one, and this is your query sequence which was 421 base pair long. Now in this you can always get your mouse there, and you can find that what that sequence is, what is the alignment, what is the accession number, and what is the E value score, which I will be explaining in a short duration. So this is our description page, where we are seeing that our query sequence has aligned to these many sequences, and there were many more. However, through the printer screen I have got only this one, this much, but when you will be doing it online, you can scroll down and see that there are many more sequences from various species to which your query sequence has aligned. So what could be the maximum score, the total score, and how much the query has been covered is given here. Now what is E value? So E value defines the number of hits which you can expect by chance. So that here it is 0, which means that all the base pairs of your query were aligning 100%, and it is not a chance event. And here you have accession numbers by which you can access this particular transcript, which is here it is like EPM2A, which is associated with, I suppose, Lafora disease, and the protein is Laforin, and it is a transcript variant X6 mRNA, and you can access these by this accession ID. So once you have that, this accession ID, then you can also look into how your query sequence was aligning to the subject sequence or to the sequence which the search engine has searched for. So here you see that your this is the query and this is the subject sequence. Your query first nucleotide is aligning with the 2393 or 2393 base pair of the subject sequence, and from there on it is aligning to till 2813 base pair. Now from here you can access the sequence. What do you find? That this, the first hit which the search engine has given, it is a protein EPM2A or Laforin, EPM2A or Laforin, and it is associated with a progressive myoclonus type of epilepsy. The below you have all the information about this entry. In what all organisms it is found in the eukaryotes, metazoa, chordata, Then there are comments about it, what genome annotation data it has been provided, for example from NCBI, the annotation status, and then as you will scroll down you will get lots of information about that. If you click on the graphics of this, you can see the alignment in the form of a graph. Here this is your query sequence and this is EPM2A and how they are aligning. You can always explore going to these arrows, or you can use the tools and tracks and whatever, you can explore all this online. Similarly, then there you can go for the FASTA sequence and you will find the sequence of the gene to which your query has aligned at the most. From here, as you can see on the right hand side, there are various ways to analyze the sequence. For example, you can run a BLAST in which this sequence of the subject which you have arrived at, that is EPM2A gene or laforin, can be uh, used to run a BLAST against all the nucleotide sequence. Similarly, you can use primers to amplify this particular gene. You can highlight the sequence features, what are the characteristic sequence features, and you can also find something in this sequence. Suppose you are looking for a particular stretch of a sequence, you can use this tool to identify. Similarly, there are articles about EPM2A gene. As you are trying to find out the gene for the linked disease, so you have come so far, so you can always look into what are the recent articles related to with this gene, what are the pathways in which this gene is involved, like for example glycogen synthesis or glucose metabolism. You can always from here link to the phenotype of the disease. Are these pathways defective in those patients in which this chromosomal region is deleted? Do you see something abrupt with glycogen metabolism in those patients? So with this you can link the information in the two. When you run BLAST using that query, now again this is just taken the accession number, as I have already described that anything you can use as a query sequence in the NCBI BLAST, and then you can again using the default as I have done here, you can run BLAST. Again the search engine takes time and thus this page appears on your screen, and then this is the way the result is again. So this is the similar result as I have described. So here these are top 109 BLAST hits on the 100 subject sequences, and this is how it is aligning. Mostly are red, almost all are red, and these are the stretch of sequences. Now these are the stretch of the sequences which were not aligning and the last part was aligning. So once you have the sequence or the all the sequence which are aligning to that gene, that was EPM2A gene, and these are the genes which are now aligning to that gene. You can select all, clicking on all, and then you can go for distance tree results. So from here you can get the phylogenetic tree of the gene of your interest. So this tree shows how the gene has evolved and how close to one or the other organism's gene is, how it is. So for example, primates are more closer to rodents and so on. So once you have the sequence, so you can also access to the protein of the sequence. You can go to PubMed and you can go on gene. From here you will write the gene which you have identified, that is EPM2A, and then you can select whatever organism you want. For example, you want the gene which is there sequenced in the humans or in the mouse or in the rat. Here the query was about a sequence which we have identified from a part of a chromosome which was deleted in the family of the affected people. So we will go for humans, and then you can have all the information about this gene in the humans, from what is the lineage, and what is the summary, what does this gene does, what all are the orthologs present. Similarly, from the on the right hand side you have all the other informations. Then you can scroll down and look into what are the other cDNA clones, 3D structures and everything, what are the ESTs associated with this gene, and so on. Now here you see on the screen that this is showing what are the transcripts present. So there are 1, 2, 3, 4, 5, 6, 7 and 8. 8 transcripts for this gene, that is EPM2A. Then on again you can look for OMIM, which is online Mendelian inheritance of man. That is, this is another search tool by you can search for EPM2A and you can see that to what all disease it is associated with. Then again you have all the related articles, then gene reference, and there is a huge amount of information already available on the internet which can help you arrive at all kind of information you are looking at, but you need to know what exactly you want to search. Similarly, when you will scroll down, you will see that there are the reference sequence of the gene. You can go to the chromosomal region from which this gene was coded, and there you can have all the information about introns, exons, what are the sequences, as the human chromosomes or human genome has been um, sequenced, aligned and deposited on internet for our ease. Similarly, there is a protein sequence and mRNA sequence. NM generally is for the mRNA sequence and NP is mainly for protein sequence, but sometimes you will also see accession IDs with XP and all, and these are for the protein. So, from here you can go to the protein sequence which this gene is coding for, and then you have all the information about the protein database. That is, you can have the conserved domain known, you can highlight some sequence features, you can use the FASTA sequence and you can run a blast as in against all the protein database and so on. So, with this we have the FASTA sequence and we can run BLAST. This protein is aligning to what all species of the protein sequences. It is aligning to the protein sequences of what all species. Similarly, you can identify conserved domains. So, if you click here, you will get to know what are the conserved domains or motifs in this protein sequence. For example, in this protein which was being coded by EPM2A, it has CBM20, it is a carbohydrate binding domain. So, as the function of EPM2A was in glycogen metabolism and in glycogen synthesis pathway, so it well goes with the function which it does, that it would bind to the starch or the glycogen. Similarly, there is another region, that is PTPC superfamily. What is PTPC superfamily? It is a protein tyrosine phosphatase which catalyzes the dephosphorylation of phosphotyrosine peptide. So, this protein which was being encoded by EPM2A, it has two domains. One is CBM20 which has ability to bind to glycogen like molecules, and similarly it has another domain, that is PTPC, which is for the which is a phosphatase domain. By that we mean that it can dephosphorylate the phosphate group from the peptides. Similarly, you can highlight the sequence features of this peptide sequence, and this is the sequence feature which is highlighted, which shows that it is the isoform B is encoded by transcript variant 2 and so on. Similarly, you can run a blast, and what do you find? That you find that again this blast would take NP as a query sequence, and using the default settings of the search engine, when we run blast, this page appears showing that it is running the job which we have assigned to it, and then you get the results. Again in a similar fashion, how well your query sequence is aligning to the subject sequence, what are the top hits, what all organisms it is aligning, all those informations we can get from here. Now you have all the sequences which has aligned to your query sequence, and from there you can select all, and then again you can go to see the distance tree results, which will bring the phylogenetic tree for this protein along the various phylums of the various taxonomic groups, for example frog, toads or turtles or insectivorous rabbits and so on. So you can see that how your protein of interest has evolved through these stages of evolution and how conserved it is. You can also select few sequences. For example, here I selected the protein sequence from the humans, then monkey, then mouse and chicken, and then you can see how well it is aligning, and you can also identify what all regions are more conserved. So you see that in these 4 groups which I have selected, mainly most of it is aligning, and only like suppose the one of the humans is not aligning to the other 3, that is monkey, mouse or and the chicken, the few last base pairs are not aligning that well. This is how this huge amount of information we just got from a few hundred base pair of the DNA sequence. So what all we arrived at, that we had few hundred base pair of the DNA sequence. From there we arrived to a gene. Then we looked into what are the features of that gene. We can look into that. What is the current literature about that? We looked into in what all species it is present, that is by using BLAST. We can also look into the how conserved that is by looking into the phylogenetic tree, in what all organisms it is there. From that gene you can go to the protein sequence. You can see into what all motifs or conserved motifs it has, how well it is conserved in the other species and so on. What is the current literature about that protein? You can look into the structure of that protein and many more. So this is the power of bioinformatics and computer science, which has given to the biologist, that when you have a small sequence, this all you can search it against all the database present. Now we will look into another search engine, that is Ensemble search engine, and very quickly we will go. We have almost understood what all we can do, but let us look into what we can do. So now we will look into another search engine, that is Ensemble, and we will go online and look into what all information can be fetched from that search engine. So this is the Ensemble home page, and you can do here BLAST or what all other tools are available. Similarly, you can have your favorite genome selected if you want the gene to be only looked into humans or in zebrafish or the mouse information. Similarly, you can go for gene expression in different tissues, how your gene expresses in the different tissues of your organism of your interest. You can compare the gene across the species, which we have also done with the help of NCBI using the phylogenetic tree which we created, selecting all the sequences. You can use your own data, or you can also upload the data which you have created. So depending on your requirement, you can search the available search engines for the information, and you can explore whatever you wish to. There are various tutorials already available on the YouTube which we can use to do so. That is all. Thank you. I hope this session has been somewhat informative for you, and which will help you in using these search engines. That's all. Thank you.